Got all the secret business out of the way.
Okay, but the speaker after this one is Hector Castro-Bastidas, who was the recipient of our Charles H. Lowe Herpetology Research Fund Award. This is going to be really wonderful. The Sierra Surutato is in northwestern Sinaloa. It falls within the Sonoran Desert region, actually the Sierra Madre. The Sierra Surutato is probably considered one of the most dangerous areas of Mexico, and because of it's the heart of the Sinaloa cartel. But Hector and his colleagues have been producing a lot of really wonderful biodiversity research in herpetofauna, and he's going to present on that in May. The second to last Monday in May, because the last Monday in May will be in New Mexico. But this will be a very good talk. And he published a paper of his results in the current Sonoran herpetologist. But tonight we have Dr. Paul Maier. He'll be speaking on Yosemite toads. Oh, all right. Someone is objecting.
You already asked the question.
Yeah.
I'm going to do it.
These toads are extremely vulnerable to ongoing climate change. The species is highly dependent upon seasonal snowpack, associated groundwater, and runoff levels. Tadpoles regularly face high desiccation mortality, exacerbated by opportunistic parasites and predators. They're sensitive to temperature-induced reduction in body fat levels that can influence their overwintering survival and fecundity. Over the past decade, they've used genomic, climate, and landscape data to elucidate how this iconic species has previously adapted alongside climate experienced during the Pleistocene and present day to predict the fate of the species. Paul and colleagues forecast how 21st century climate change will encourage range shifts while simultaneously favoring the adaptive success of certain lineages, offering practical conservation suggestions from their novel landscape genomic approach. Dr. Maier is the lead population geneticist at Family Tree DNA and Gene by Gene, where he builds ancestry estimation tools and studies the evolutionary history of human life on Earth. Since 2018, he's developed numerous products and features. He earned his PhD in evolutionary biology, studying the genetic past, present, and future of a much squishier creature, the Yosemite toad in the Sierra Nevada of California. So let's welcome Dr. Maier.
All right. Well, thank you, Robert. And hi, everyone. It's wonderful to be here at THS. These days, I mostly talk to genealogy crowds about genetics. So it's kind of nice to be, talking about frogs and toads again. I'm really just a big kid who likes to chase frogs around. So I'm sure most of you guys feel the same way. So you guys are my kind of people. Since I finished my PhD 5 or 6 years ago, I transitioned to working on human genetics, as Robert said. So I'm the geneticist for Family Tree DNA, and I have been for the last 5 years. Today's talk will summarize a previous chapter in my life. Over 10 years of research on the Yosemite toad. I think we're up to closer to 15 now. What we've learned, hopefully some lessons for you guys if you're studying different herps in your own respective systems. So some lessons that might be applicable. And we've published a lot recently, so I think it's a good time for a big picture talk. And for the record, while I was snapping this photo, my wife said, there's bird shit right there. And I said, yeah, no one's ever going to see this picture. Now it's on the cover of a talk. Yeah, now it's on the cover of a journal. So everyone gets to see that now forever. And by the way, my beautiful wife and daughter are here. My daughter, Junie, just turned 7 months today, little tadpole. So she might have some questions, and if she's a little loud, she might step out for a minute or two. So a little background on me. The Sierra Nevada will be the backdrop for this talk. I've been mountaineering in them since I was about 5, so I guess going on like 35 years now. So it's like home for me. And like many people in this room, I love nature, I love critters, and I love solving puzzles, and that's really kind of what dragged me into genetics. So my core research areas are evolutionary genomics, popgen, of course, phylogenetics, which is estimating or reconstructing trees, phylogeography, which is kind of combining trees and maps together, and of course, herpetology, and these days mostly human evolution. And since I joined Family Tree DNA about 6 years ago, I've used some of the published work that even I'll talk about tonight to build some of those tools. So this is one of those tools that I've built called Globe Trekker, which traces over 50,000 individual human migration paths across the globe. Everything you see here, including the map and the exposed land masses over the last glacial maximum and the ice sheets, all that is built from scratch. And yeah, this is, I mean, I'm basically a biologist who got out of control, I think. So you can see how the paths go through Beringia, and as the glaciers recede, there are these two paths. There's the Pacific path and the inland path, and then it goes down into Central America, and this clade spreads out into both North and South America.
Oh, what is this big trench across the earth? So it's
Oh, these are sea currents, and they're
Okay.
actually modern day sea currents, so it's a little misleading. Of course, they changed a lot over time, but it gives you some idea of where the water is flowing, directionality. We actually use that to infer the paths that go across the ocean when necessary. So,
That's very cool.
so anyway, ice ages are kind of relevant for this talk. And one order of business, Robert already brought this up, but I'm involved in a large-scale genomic study on Gila monsters, so please reach out to me if you do find one. You can take a screenshot of this or a picture so you don't lose my email. And this particular monster is special because we found it on our property shortly after moving here a couple of years ago, so we call it our landlord monster. So let's take a couple minutes to transport ourselves into the Sierra Nevada, where this story takes place. Here you see my hiking partner, Dr. Danny Nielsen. And you know, for 5 billion years, tectonic forces and beautiful glaciers have sculpted this playground of granite. And it's an incredibly rugged landscape filled with glacial U-shaped valleys and jagged peaks and garden meadows that so inspired John Muir's interest and respect. 10,000 lakes, 20,000 meadows dot the landscape. And in the midst of this crazy ruggedness, somehow pops a squishy little toad. So how to get there and why and how to survive? Well, with a team of USGS employees and interns and volunteers, some of whom I think are listening from home. Hi, Amy. We collected just a massive number of samples. So these are Yosemite toad alpine meadows or subalpine meadows. These are places they actually breed. And then as we advance here, you're going to see some montane meadows at the lower elevations of where they exist. This is for 6,000 to 8,000 feet in the red fir belt. So this gives you a sense of the sites that we're talking about. And this is what actual Yosemite toads look like, males and females in plexus. So males are a little more drab color. They tend to be kind of olive or even yellow. They're brighter colored during the mating season. Otherwise, maybe brown, kind of mottled or more boring. Females are this beautiful reticulated pattern, kind of this ruddy color on a dark background with these cream borders. And so they're the most dichromatic, sexually dichromatic neuron in North America. And so females are very starkly different from males. Males, as I said, are kind of this more plain.
Any way we can crank the lights down a little bit?
All right, let me take that. But some are more melanistic, kind of more dark colored like this. See better? Oh, that's great. Subadults are more similar in appearance. Metamorphs look like tadpoles with legs, and tadpoles are this very dark melanistic coloration because they're trying to soak up as much sunlight as possible before the breeding season is over, which is very quick. All right, so that gives you some sense of the system that we're talking about, because I know you guys are, thinking Sonoran Desert all the time. This is a very different system. Before we continue, as a genealogist, I think it's interesting to learn the academic pedigree of researchers who taught us the most about a species. Okay, yeah. So in this case, there were two, Ernie Karlstrom and Cynthia Sherman. So what you're seeing here is the ancestor academically. So we have Louis Agassiz taught Burt Wilder at Cornell, who taught Albert Wright, who eventually taught Raymond Coles, the famous Raymond Coles, who was the advisor for Robert Stebbins. I think everyone knows him. You have a Peterson guide. And he was the advisor to Ernie Karlstrom. One of his early students was Ernie Karlstrom, who was one of the first people to teach us about Yosemite toads, one of the first world experts, got his PhD in 1956. And the other person is Cynthia Caggery Sherman, who studied under Richard Alexander at UMichigan. And fun fact, Brad Schaefer is also the academic grandson of Alexander. So everything's kind of connected, small world. But Cynthia taught us more about the toad's natural history, I think, than any one person before or since. Let's also glance at where canorus, species we're talking about, fits into the tree of Anaxyrus toads. So this is the genus that includes pretty much all North American toads, except of course for the beloved Sonoran desert toad that we all know and love here, and the Gulf Coast toad in Texas, and cane toads if they make it up in Texas, but pretty much all North American toads. So canorus is kind of the high elevation isolate of the Boreas group down here. And the Boreas group, they're the outgroup to the rest of Anaxyrus. I don't know if I have time to go into it, but some of my former mentors and advisors found that there actually were Boreas populations that looked a lot like canorus. There were some in Montana and Alberta, Canada. Okay, so I'm afraid this talk is a little bleak at times. It's about climate change and adaptation and picking winners and losers. But it doesn't mean identifying like, I don't know, the Lannister toad will kill the Targaryen toad. I never watched Game of Thrones, so I don't even know if that joke makes sense. But it simply means accepting death as a part of natural selection and trying to shepherd the species into the next century. So without further ado, let's get into the meat and potatoes of this talk. Anthropogenic climate change is rapidly reorganizing our natural world, as everyone here knows. More than a third of species have moved. Calcium-containing critters are literally dissolving in oceans. Plants are flowering at new times. They're losing pollinators, and amphibians are breeding earlier. And up to a third of species could be on the path to extinction by 2050. Those vertebrates most at risk, not surprisingly, endemics, specialists, amphibians. For example, over 2/3 of the stream-dwelling harlequin toads, genus Atelopus, are listed either as extinct today or as critically endangered. Yosemite toads are particularly sensitive to climate change, as they are montane and subalpine dwelling and rely on extremely shallow snow melt pools, where larval ponds tend to be less than 5 centimeters deep. They dry up very early in the summer. And this may give them a competitive edge against other amphibians, at least that's my hypothesis for a while, but it leads to significant mortality. And previous studies have estimated that Yosemite toads have disappeared from up to half of their historic sites. And there's a little caveat in there I can maybe address in the Q&A later. But regardless, they exist in very low abundance everywhere you find them. And because of the specialization, they're endemic to this very small patch of the Sierra Nevada, above 6,000 feet. And if I zoom in here, most of my research focuses on the 2 national parks, Yosemite and Kings Canyon. To help orient you, here is Lake Tahoe up the north there, Mono Lake, just east of Yosemite, and this is all the foothills to the west of the Sierra. So zooming in farther, we can see that their meadow habitat is very patchy. So here we have all meadows. And here are the toad-occupied meadows only. So let's zoom in on this one representative meadow down in the bottom right. This is a fairly small toad meadow. It's only about 200 meters across, and it shows that their breeding habitat is only a small portion of the total area. And to maximize their season, they breed as soon as possible when meadows are still about half covered in snow. It's crazy to see toads hopping over snowbanks. They choose extremely shallow pools, as I've said several times. And it's very common now to find entire meadows filled with dead cohorts of tadpoles. All right, so now that I've scared the crap out of you about climate change, how bad is it for the Sierra Nevada? Well, the expected effect, this is from a UCLA report in 2018, so it's a little old now, but the expected effects include a 7 degrees Celsius rise in springtime temperature. Wow, that's a lot. 2/3 reduction in snowpack volume. I mean, the entire ecosystem is kind of driven by snow melt, so that's quite a bit. 50-day earlier snow runoff or melt-off date. I mean, for the subalpine areas, that's most of the season. That's, they have about 2 months only. And the largest changes are expected at the mid elevations, kind of that red fur belt, 5,000 to 8,000 feet. There have been other incidental reports that are scary, like I saw an NPR story in 2020 about zombie forests, where there are these forests that are technically still standing, but there's just, one disturbance event, a fire, something like that, a disease outbreak, and they're gone forever. So for these reasons, toads are a model species for specialist amphibians threatened by climate change. And a conservation genetic study on toads can be helpful for other species that are experiencing similar climate pressures. So let's talk then about conservation genetics. What's the goal? On the surface, it's about conserving populations of organisms. But as Darwin realized, and others have since reaffirmed, populations are really just lineages at one point in time. So, I'll talk about that more, but if there really is this temporal element of it, this flow, then which timeline matters the most? Well, we could use phylogenetics to focus on the ancient past, and lots of people do this. Lineages also evolve by something called lineage fusion, and I'll talk about that more, but basically it's two lineages hybridizing for mutual advantage. So maybe if this is the case and there's some conservation implication, we should focus on that in the recent past. Or perhaps we should focus on present day populations. We're kind of biased by our present day perspective, right? So it's very tempting. Present day population diversity certainly has a lot of importance. It's kind of fuel for future lineages. But ultimately, conservation is about conserving species into the future, shepherding them into the future, making sure that they don't go extinct. And we often try to extrapolate from the present and the past into the future, but that's not so trivial. The future may throw a curveball. The trajectory under climate change, for example, may not reflect the past. So without explicitly modeling the future environment or the ability for populations to adapt and change, we have poor predictability. So to recap, lineages are just populations moving through time, and we're biased by our present day perspective. Future resiliency depends on past and present ecology and evolution, and future environmental change as well. And so ask yourself the next time you read a conservation genetics paper, what's rushing over here? What's the time frame that's in focus? What time frame of these four is the focus? So for example, here's a study on black salamanders that used phylogenetic methods. They discovered cryptic Pleistocene diversification. And again, there's no guarantee that past adaptations have any relevance to the future. Here's a study that I think a lot of people in California will immediately recognize based on the picture. So you have this hybridization between an invasive tiger salamander and the native one. And so hybrid vigor may indeed have some future relevance because it introduces a huge amount of new genetic variation that can later be selected on. Well, here's a study on the Columbia spotted frog that looked at present day patterns, dispersal, gene flow, and they described population structure and dispersal habitat to maintain genetic diversity. So connectivity is super important for kind of maintaining that diversity that can fuel future adaptation. But the ultimate time frame, again, for conservation is the future, and it's also the most difficult to predict. This study on poplars forecast where future diversification will take place using temperature and circadian rhythm genes. So they really try to do this. They explicitly model the future using genomics. The ideal conservation genetic study would integrate aspects of all these studies and model future outcomes. As a 20th century biologist once said, your future is whatever you make it. So we should try to make it a good one. All right, so let's take this back to the future kind of paradigm and apply it to Yosemite toads. So I'm going to quickly summarize 4 chapters of my research for these 4 different time frames. All right, so chapter 1, aka what happened in the ancient past and why is it relevant now? The alpine zone is a fleeting environment. And by the way, this is a beautiful snapshot of the Eastern Sierra with the rain shadow in focus, which is gorgeous. So the alpine zone is very fleeting, and yet many plant and animal species make it their home. So what happens to them once glaciers advance? Well, after all, it's happened not once, but countless times as Earth has cooled off during the last 3 million years. Here you see the last 8 ice ages over the last 1 million years. Ice ages occur about every 100,000 years or so, and previously it was every 41,000 years as there was more carbon dioxide in the atmosphere. And there's plenty of evidence that glaciers crank out new species. The idea is that advancing ice sheets squeeze mountain-dwelling organisms down into refugia, where they slowly adapt in isolation. This is New Zealand, by the way. So there's plenty of evidence that organisms show similar lineage breaks because they inhabit the same refugia. So this would be evidence that refugial habitat is a strong influence on shaping lineages. Alternatively, you might think that an elevational gradient like this mountain range is what leads to speciation. And this is often called ecological speciation, right? So there are examples of this in Sierra Nevada plants and various frogs around the world, like Indian bush frogs and poison dart frogs. Well, for yotoads, we predicted that both are important. We call this the glacial pulse model. So it begins with prototoads. I trademark that term. Prototoads kind of make their way upslope in this white polygon here, and they slowly adapt over that climb and become kind of a gradient of white and gray adaptations. And the glaciers force it into two refugia on west and east sides of the mountains. Glacial refugia then reinforce those differences. And then you have two lineages. You have, after the glaciers recede, you have these two lineages, white and gray. And this can continue many times until all niche space has been divided up amongst new lineages. In our system, you can imagine this being low elevation could be Boreas, which is the western toad, and then you have two lineages of Yosemite toads, low and high. So the predictions of this are that the timing of divergence should relate to glaciers in some way, that the ecology of divergence should also relate to glacial events, and that the size of secondary contact zones depends on how similar the niches of those two lineages. So in other words, if you have two lineages that are very similar ecologically, their hybrid zone should be wider because they're more able to interbreed. All right, so what did we find? We found that there are four main lineages in Yosemite. So conveniently, they're north, east, west, and south. So north and east are kind of the, yeah, sweet. North and east are the kind of high elevation lineages in red and blue, and black and green are the low elevation western and southern lineages. So we found these four lineages, and you can see the ice sheets in these vertical stripes on the tree on the left there. They correspond very closely to the major lineage breaks. And even if we zoom out to show the entire species, this is both national parks now, there's this 2 million year old glacial event that may have helped spawn the entire species. So this is consistent. Niche diversification also likely did reinforce this pattern. So each pair of lineages experienced opposite ecological pressures on each side of the mountain range. You have a split on the tree, you have a split between, I'll just call it by color, between blue and black and between blue and green, and each of those lineages experienced opposite pressures on both sides of the mountain range. We even found a genetic signal, a spatial genetic signal, in the data suggesting that the high elevation lineages recolonized from the east. So you see these white arrows in the top right. There's a kind of a spatial gradient indicating that there's recolonization from the Owen Valley to the east back into the mountain range. And you might ask, why don't you get the same thing for the black and the green? Well, because look at where the ice sheets are. There was no need to move. So they kind of, their refugia was their current distribution. They didn't have to move at all. So this is all very consistent. And you'd think, normally you'd think that genetically distinct populations or lineages mix the least, right? I mean, this is what speciation is. The more distant they are, the less able they are to produce viable offspring. Well, nope. The amount of secondary admixture correlates not with time since divergence, but how ecologically similar they are. So the red and blue are the most distant lineages that have a hybrid zone, but because they're both high elevation, their hybrid zone is the widest. So kind of checks all down the list of predictions. We were very happy to publish this work in Evolution a few years ago. And so just to kind of recap, so the timing of lineage formation is consistent with glacial barriers. Climate played an important role, and twice there was a split between high and low elevation lineages. It looks like refugia helped reinforce that pattern. And then secondary admixture correlates most with niche divergence. Pretty cool. All right, so now let's briefly discuss mixing between lineages, which occurred in the more recent past, AKA what happens if I breed with the wrong individual. All right, so there are many papers now describing so-called lineage fusion. And lineage fusion, again, is briefly, it's two lineages coming together and hybridizing for mutual advantage and then persisting through time. Leading to fantastic paper titles such as Mating with the Wrong Species Can Be So Right. And so in the interest of time, I'm just going to give the bullet points. But aside from our four main lineages, we found these three fused lineages. And they're found in the north and south contact zone. So we had two fused lineages in pink and purple on the map up here. And then this kind of cyan color is another fused lineage down in the southern context. So again, these are lineages with dual ancestry. And just forewarning to set your expectations, this small part of the talk is a bit technical. I'm going to do my best to keep it very big picture and simple, and I have some pretty graphics, but just stay with me and it'll be toads and pretty pictures and meadows after that. So it's worth it, trust me. All right. So this is important because their dual ancestry gives them the highest diversity in the park. And this recombinant diversity just means that there's been hybridization and there's been recombination between two different genetic backgrounds, and therefore the diversity includes those backgrounds in some new combination. So recombinant diversity may fuel future adaptation to conditions never seen before. And this is a process called adaptive introgression. So let's see how this works. So we have these red and blue hypothetical toad populations, and they separate from each other, and eventually they hybridize. And those purple hybrid toads offer new genetic material never seen before. And some of those new alleles spread, whichever direction is most beneficial. So in this case, they spread to the right into the blue lineage because that blue genetic background offers the greatest advantage to those toads. All right, so that's adaptive introgression. There are countless examples of this in nature, and more every day. So briefly, there's the hybrid zone between Ambystoma species in California, where there's clearly certain viability genes or size genes that are actually making their way through California faster than other genes. There's an example in the Galapagos with the median ground tanagers, formerly called ground finches. Heliconius butterflies essentially reshuffle the genetic deck to trade optics genes, which confer color and pattern diversity. So they have these adaptive radiations by sharing genes. Even in humans, Neanderthals contributed immunity genes and hair and keratin genes. Even Tibetan populations of humans carry a certain gene that confers hypoxia adaptation. So they're able to withstand lower oxygen levels, and that's been contributed not from other humans, but from the Denisovans over 40,000 years ago. So this is a fairly common thing. So we wanted to look for evidence of this, and we also saw an opportunity. We have 3 naturally occurring hybrid zones, meaning 3 kind of statistical replicates for testing how predictable nature is. So this is simplified, but we had kind of 3 big questions. One, is there evidence for first adaptive divergence, followed by adaptive integration? 2, how predictable is adaptation? And I'll define what I mean by predictable in a second. And then is there any relevance to tadpole growth and development, maybe as it relates to climate change? Okay. So did we find evidence for adaptive integration? Yes. If you look at the right there, those plots kind of show the few genes that are pushing directionally from one lineage into the next. So for example, the red lines at the top right show genes that are pushing into the red area from that hybrid zone. And every hybrid zone showed some of this pattern. Now, did we find any predictability in the adaptation process? To answer that question, let's first break down the process a bit further. So what happens when 2 species or populations diverge? Humans and chimps still share, about 99% of their DNA. So we don't completely change 100% overnight. It's a slow process, and it usually happens with these small islands of DNA getting bigger and bigger, usually accruing adaptations to different environments over time. So we call these islands or genomic islands. And the most extreme of these tend to be the most prominent adaptations. And so you can see this in the toad chromosomes, these hypothetical toad chromosomes, the red and blue, specific adaptations grow bigger and bigger over time as they stay isolated. All right, so we did find some predictability. Some of these genes predictably would introgress. So these red and blue toads have red and blue adaptations, right? One of them is moving from red to blue, shown by the purple here. The purple, again, is kind of this recombinant hybrid allele that's been created in the hybrid zone, and it moves from the hybrid zone to the right into that blue lineage. And we found some of this in every hybrid zone, hence the predictability. We also found a different layer of predictability. So when you consider adaptive divergence, those genes that are kind of promoting speciation, the question would arise, is it the same genes always causing speciation? And the short answer is, what we found is no, it's different genes, but it's the same specific functions. So you kind of have these different gene complexes doing the same stuff in each hybrid zone. And this is a cool example of parallel adaptation. All right, finally, we found these 10 genetic loci underpinning tadpole growth and development. And for example, we found a gene called LPIN3, which is crucial for lipid metabolism. And it's probably very important if you're a tadpole and your pond is slowly, inch by inch, getting shallower every day. And we found that it's a speciation gene in every of the 3 hybrid zones. And it's also introgressing, adaptively introgressing from the east to the west. I actually just published this paper in molecular ecology this month. So this is my 5th and final PhD chapter. Woohoo! Finally stoked to move on and move on to bigger, better things, but I'm also very satisfied to be, contributing a small part to this species' natural history. So to recap before moving on, adaptive introgression is an underappreciated source of novel diversity for the species. More broadly, we tested some basic questions about adaptation in nature, and we found 2 different forms of predictability. It's pretty cool. And one example of this is relevant for toads, this lipid metabolism gene, which is a possible adaptation to desiccation resistance. All right, so let's move on to the present day. AKA, how fast is your transportation? My wife helped me Photoshop a toad in a DeLorean. Going precisely 88 miles per hour. All right, so there are 2,500 meadows in Yosemite. And they're spread across just 3% of the landscape. So they're kind of like islands of habitat. And toads occupy just 10% of that, so 0.3% of the landscape. And here's one toad meadow. This is in the White Wolf area, which is western Yosemite. You can see it's part of a toad meadow neighborhood, which I'm defining just as a cluster of nearby meadows. Okay. And there are many neighborhoods or meadow neighborhoods in Yosemite's western lineage. So this is kind of like a galaxies within galaxies kind of setup. So we wanted to understand, number one, what's the boundary of a population? If you ask me a question about a toad population, I could ask you, what do you mean by population? Like, where do I draw the line? So we wanted to understand that. And number two, we wanted to understand what keeps them connected. Because kind of, like I've said before, connectivity is kind of the glue that keeps together populations so that they can have enough diversity.
Este es interesante,
adaptations.
Quirino.
So first, we formalized the idea of meadow neighborhood. Genetically, we found 28 of them. So each of the colors in those pie charts is one of 28 different meadow neighborhoods. And we found that, by and large, meadows themselves, not the neighborhoods, are consistent with population boundaries. Not that the neighborhoods and the lineages are not important, just that when you're talking about present-day ecological patterns, we should be talking about meadows as kind of the unit. So they explain population structure 6 times better than neighborhoods. But that's not the end of the story. We found these, what we're calling hub and satellite meadows. Hubs are these just beautiful, big, lush, central, spatially central meadows that toads tend to hang out in the most. This is Ireland Lake for anyone who's been to Yosemite before. It's kind of eastern Yosemite. So it's this big golf coursey meadow that's next to this big, beautiful lake and has all these peripheral meadows within a few miles of it. So in this case, we have, as I said, kind of one big fat meadow in the middle, and that's a hub, and toads spend most of their time there. And we found that genetically, toad migration tends to be from satellites into hubs, where they spend most of their time and perhaps have the highest fitness as well. Environmentally, we can separate out hub and satellite meadows 83% of the time. So hubs, statistically, the 3 best predictors are that they're larger, they're flatter, they're topographically more simple, and they tend to have more mappable water bodies within a kilometer. We published this in Frontiers. And we also used landscape genetics to understand their migration corridors better. So which environmental features most facilitate DNA moving across the landscape? So we invented some new methods I'm not going to go into and selected some environmental features from 5 broad categories: climate, vegetation, moisture, topography, and disturbance would be things like fire perimeters and trails as barriers. And for every toad corridor, such as this one, we also considered the bandwidth of environmental influence. So this would be a narrow bandwidth, only considering kind of the direct path from A to B after kind of correcting for the most, sane path that they would actually take. Here's a wider bandwidth, and then here's the widest bandwidth, really integrating all possible paths between A and B, even the ones that are probably used much less, but kind of normalizing for how much they're being used. Okay, so what did we find? We found that snowpack variability and associated runoff and recharge have the biggest effect for connecting toads between meadows, but also effects from southern slopes and probably the associated heat that goes along with it, rocky and shrubby corridors, which is a little surprising. They tend to be more xeric, but they seem to be important in certain places. Trails did come out. There was a signal of trails being barriers. And that's just habitat between meadows, what about habitat at the meadows themselves that either cause toads to decide to emigrate or not, or immigrate into them or not? So there we found that hydrology, different types of specific hydrology are important, as well as willows, which are often used for cover and for hibernation. Adult toads, especially females, live a long time, 15 upwards of 20 years, potentially migrating long distances. So researchers always kind of lament how it's impossible to study them once they're gone in the upland environment. So it's pretty cool to have some sense from this genetic data set of what they're doing most of their time, especially the females, as they're in between this 0.3% of the landscape. All right, this work made the cover of Heredity. This is now a celebrity toad and celebrity birdship.
Yeah.
So to recap, meadows are highly distinct clusters of meadows that we call neighborhoods, have this hub and satellite structure. Hubs tend to be larger, wetter, and flatter. And we identified toad migration corridors, which environmentally tend to have more, less snowpack variability and associated recharge. And willows and rock fields and trails also play a role. All right, so the last chapter, what does this all mean for the future? That's kind of the most important part, right? AKA, how screwed are we? Okay, so toads have four possible responses to climate change. They can adapt to it, they can move, they can try to buffer its effects, or they can sadly die. Or as my colleague Alexa once put it, from glad poles to sad poles. But that's what we want to avoid, right? So in our Heredity paper, we predicted a massive range shift upslope from kind of the montane environment up into the Tioga or the eastern lineage. And zooming in, you can see that low elevation toads will be forced the most. The red, by the way, connotes the most amount of migration. So you have the most migration down in the south, moderate amount in the west, and you can see that there's a lot of movement upslope into the high elevation lineage in the east. Okay, so perhaps they'll move somewhat, but they may have limited time, right? This is happening quickly. So what about adaptation? Well, adaptation depends on kind of roughly supply and demand. How much supply of climate-associated diversity is available, and how much climate change, how much climate pressure will exist? This is where we have to pick, sadly, winners and losers. And remember that this is the most crucial question. We care about lineages in the future, not the past. So future toads will be shaped by local climate, and they'll survive only if they have enough climate-associated diversity to overcome climate change. So briefly, here are the steps for predicting the future. We have to identify genetic loci that are affected by climate in some way. We have to create a map that shows regions where toads are under similar climate pressure. And then based on those selection pressures, we have to simulate their fitness, right? If we know the selection, that's one part of the equation, but we have to know how they actually respond to that selection. So who's going to survive the next century? We found 24 genetic loci, including 4 known genes associated with climate adaptation. And the genes are kind of interesting, so I'll just rattle them off here. We have the first one, MAP3K5, is a protein known to mitigate climate stress. So it kind of acts along with heat shock proteins to protect amphibians during extreme temperature fluctuations. We have the TTPA gene. Tadpoles use it to secrete vitamin E, which expedites their development, which is important if they're in desiccating ponds. The PinLip one has to do with innate immunity in embryos. And the KIF1C gene, we know the least about, but it probably has to do with embryonic development as well. So we have these 24 genes, 24 loci, and here they are shown as a PCA plot where colors represent similar environmental adaptations. Colors are similar environmental adaptations. Okay. Environmentally, toads fell into one of 3 groups. We have high summer rainfall, high snowpack, or neither. So let's look at this on a map. The north will experience high snowpack and runoff, and longer hydro period may be beneficial for those tadpoles so that they can grow larger. They're accruing more biomass. The east will experience high summer rainfall, which is probably where we have faster drying pools, so they're developing more quickly, and the summer rainfall may help them kind of bridge that gap if they don't have quite enough time. And the low elevation toads will experience neither, not high snowpack, not high summer rainfall. We think this may have something to do with the phenological wholesale difference where they're relying more on winter precipitation. And so they kind of just have an earlier breeding season. And this makes a little sense because we find the largest tadpoles lower down. It could be that they're adapted to those higher temperatures where everything's happening very early, but things get hot. And on the right here, I've simply done the same thing. I've made the same map, but I've grouped them into conservation units. Okay, so let's look at the east here. They have the most climate-associated diversity. That's the bright colors. And they also have the least amount of climate change expected. That's the blue colors in the second map. So the most diversity associated with climate and the least selection pressure from climate. Overall, we predicted a almost 30% decline over the next century. That's a lot. And due to differing evolutionary trajectories, the eastern lineage is most likely to persist into the future. Their fitness will decline too, obviously, but will decline less. So this is a climate refugia. We've identified 3 future lineages, identified the east, or we call it the Tioga lineage, as a special place. So it's it has friendlier levels of natural selection, it has higher adaptive potential, and as I showed earlier, it's going to be a destination if there are any range shifts. And then as I showed in part 2 of the talk, it's also a source of adaptive introgression. So some of those beneficial genes are moving out of the east into other places as well. We published this in Evolutionary Applications. So in conclusion, there are 4 distinct types of conservation units, each representing a different temporal process. Pleistocene era lineages show toads can and did adapt to climate. Recent lineage fusion gives us a novel source of adaptive potential. Interracial toads may be the future. And those contact zones are special because they're like genetic lines of communication that we need to keep open. Modern day population boundaries show us where the glue is for keeping lineages intact, and protecting them is important to protect that connectivity. But ultimately, these 3 future lineages are what to expect in the future. The East is special because it has the most, as I said, potential as well as the least pressure. The low elevation lineage we could actually combine, although it's 2 clades historically. I think that combining them in a management sense could help them a lot because each of them has kind of a lower effective population size, and if they share genes, they may actually be able to persist longer, given that they're under the same selection pressures anyway. And the looming question is, how does the fungal pathogen BD play into all of this? But that's a story for another day. All right, I'd like to thank all these beautiful, fine people and institutions who helped out over the last 15 years and made all of this possible. And a special thanks again to Ernie Karlstrom and Cynthia Sherman, who gave us so much knowledge. And with that, I'd love to take any of your questions. Thank you.
Did you have any positive BD up there?
In Yosemite, yeah. Actually, in Kings Canyon, we sampled for BD in 2011 and found nothing. But the Sierra yellow-legged frog, we found lots of positive samples in the same watersheds. So I think that might just be an artifact or a sampling issue, but it's definitely there. And there was a grad student back in, I think, 2012, Celeste Dodge, who found that there's a correlation between museum specimen BD positive results and the decline, happened around the same time. It was like the late 90s, and that's when you find the most BD positive samples. So there's definitely correlational evidence that it's there. Yeah.
I guess that's why they call this the tip-toed frog or the tip-toed toad.
The tiptoeing toad, that's
Yeah.
right, yeah. They keep their body up above the snow to kind of keep their body a little bit warmer.
Okay. So the definition of migration, if we're looking at, sometimes you look at migration in kind of a short distance. An adult that's going in a certain area, like going into a rodent hole and to overwinter, and then come back to a breeding pond. So that's a short distance movement. So that's not really migration. We're talking about migration in terms of time and distance. Migration is over generations or and talking about not just hundreds of meters, but miles.
That's right. Yeah, that's a very good question and good point. So in genetics, this is kind of assumed or known, but outside people kind of use different terms. So dispersal is what ecologist would call migration, perhaps. Dispersal is just movement from point A to point B over any time frame you're interested in. Migration in genetics means moving and then interbreeding with that population and leaving genes in the next generation. So leaving some progeny. And yeah, it can't take many generations in the case of the species because they're taking, 5, who knows how many years in some cases to get from point A to point B.
How long, what's the metamorphosis time?
It takes about 6 to 8 weeks. I've seen as little as 5.
Okay,
So they're,
that's long.
I mean, that's relatively short. I mean, there are definitely spadefoots who are doing it in an insanely short amount of time, but for toads, I mean, it's only short, 4 to 6 weeks sometimes, and then upwards of 8.
I recall from a long time ago, some studies associating increasing UV radiation with some of the declines.
Yeah.
For them, I'm just curious with your 25 genes that you found that were adaptive in that round, were there any related to protective elements like that?
Didn't see anything about UV specifically. It's been a long time. I think there was one gene that had something to do with, I'm going to botch it now because I don't remember, but something to do with protection from the sun broadly. And it wasn't a major signal in the data. So that has been studied earlier, whether UV or pesticides or these other exogenous factors are important for them. And nothing's really come out as a strong signal as it has for the yellow-legged frog. For the frog, it's, fish and disease. For the toad, there is a clear indicator that disease does induce morbidity and mortality. But at the population level and in the wild, it's still not clear whether or how much that's an effect. And this is probably the best evidence we have that climate plays a role, has an effect. And it's all interesting, but there's no smoking gun causation still. So, yeah.
Maybe I missed it somewhere along the way, but so how is the rate of adaptation or evolution relate to climate change? Because I guess the rate of climate change is much quicker than historically it had been.
Right. So the question for people listening, I should be repeating this. The question is, how does the rate of climate change compare to the rate of adaptation? I mean, we don't actually know how fast they're adapting. We just know how fast they would have to adapt in order to outpace climate change. And I mean, it's a great question. I can only speculate, but the effective population sizes that I was getting were something like 20. And that's not actual individuals. That's in a genetic sense, 20 effective individuals that are leaving progeny. So that's a very small number. And a very small number means that adaptation is very slow. It takes a lot of individuals for adaptation to happen fast. So I don't know, but I would imagine quite slowly. 100 years is not much time. Yeah.
What are the temperatures in these ponds when the eggs are being laid and the tadpoles are developing?
In the early season, when it's all snow, around 15 degrees Celsius. And by the end of the season in July, August, depending on elevation, I've recorded temperatures close to 35 degrees Celsius. It's baking.
So does the shallowness of the water help them warm up faster during the day?
Yeah, that's the idea. And they're more melanistic than other species, and even lower population, like higher populations have more melanin than lower populations. So that's the thought, yeah. Yeah.
You might have said this already, but does the temperature of the water affect the rate of development?
That's, yeah. I haven't done any of that work myself, but there was a physiologist who worked on that in the 50s. And I think that's what that paper says, is that, yeah, there is a correlation there.
Is their sex dependent on temperature or?
That's a great, so the question is there sex dependent on temperature? The official story is no, but I've always wondered because there was a study in the 1920s that looked at bitters organs. Bitters organs are, all toads have them. And I'm going to forget the details now because I haven't read this in a long time, but there are toads that do change sex and or have sex dependent determination in Europe, like the green, I think there are populations specific, the green toad, I can't remember, where the bitters organ plays a role in it. Yeah, I don't remember all the details, but Yosemite toads had the largest bitter organ of any individual examined in that study. And so I've always wondered, is there something going on, because they're the most sexually dichromatic species of anurin in North America. And is there anything going on with sex determination? Do they have maybe temperature dependent sex determination lower down and genetic determination higher up? I don't know. But it's really hard to study these guys now because they're federally threatened and you can't really get permits to do any kind of experimental work, but I would love to look at them, yeah.
So we have these three really major big populations. One on the east is the one that's going to be the ones in the future. Then you have the two others, but you were talking initially about hybrid vigor, hybridization being a source of diversity, genetic diversity. Are we going to lose that because there's more separation and less overlap and less hybrids that are there to cause that diversity?
Lose some of the hybrid diversity or just
Yeah,
because,
because you have more isolation of those three large populations.
oh, so are we going to lose diversity over time because of the isolation? Well, if it weren't for humans, I think we would get the same cycle over and over. There would be a loss of diversity over that, whatever it is, 100,000 year cycle. And then you would have secondary contact again, which would reintroduce the diversity and the whole process would repeat. I think now, not only are we not likely to go back into one of those cycles again, who knows if they're going to persist, but each population is itself more isolated. So populations are blinking out or becoming more isolated. So I think that's kind of the biggest factor is just everything is becoming isolated because of that extirpation. The whole species is just becoming lower and lower abundance. So that's the major loss of diversity now.
This is kind of a loaded question, but you've obviously shown that hybridization between lineages can be adaptive and beneficial, and even showing, one-way introgression of those adaptive genes. Now with the ability of us to do genetic rescue, would you have human mitigated hybridization, like population mixing, or do you feel that it's best left naturally to maintain diversity and connectivity and let it go from there?
Okay, so Taylor asked his own words, a loaded question about should we consider doing artificial hybridization in order to kind of amp up their diversity? Is that a good thing or not? Personally, I'm okay with the idea because the same sort of thing happens naturally anyway. I think there are kind of degrees of how responsible or not responsible you want to consider yourself to be. So, I mean, if you take individuals from all over the park or all over the whole species range and, just kind of combine them, I think that's probably a little irresponsible. But if you assist a process that's naturally occurring, if you, find two lineages that you think would hybridize normally, but they can't because of us, I don't see any problem with that. And I think that would help a lot. I would love to have more experimental work done before just kind of blindly going into that. We just finally were able to breed them in captivity at the San Francisco Zoo for the first time recently. And people have been trying that for decades. So, I mean, that's step one, I think. And some more experimental work, I think, should be done before we start introducing artificial hybrids.
Knowing how readily toads hybridize between species.
Right.
Do you see a potential situation where during climate migration, other species might enter the range of boreal toad?
Oh, of the boreal toad.
I mean, excuse me, of
canorus?
the Yosemite toad. Sorry.
Okay, yeah, the question from Robert is it possible that other species, other species besides the western toad would enter the range of either the western toad or the Yosemite toad?
Yeah.
I'm not sure what that would be. The only thing that's close is the black toad, and they're a genetic isolate. They're not going to help much. They're an endemic themselves. So, and the woodhouse toad is so far removed. That's the next closest thing I could think of. Yeah, I don't know that would necessarily help. I know Dr. Bufo in Texas in the 50s and 60s did a bunch of artificial hybridization experiments. And I'm sure it's possible to stick a woodhouse toad next to canorus and, maybe 1 out of 100 times that would work, but I don't think that would necessarily be beneficial. Too far removed. Yeah.
So are there any other inerns or uridels that are sympatric that share that space? Any hylids or pseudacres?
Yeah, Pseudacris regilla. I think for a while they had it separated into Pseudacris sierra, and I think they've gone back. They're always splitting and lumping, so I don't know. But yeah, Pseudacris, whatever it is, regilla is overlapping with them, and that's probably the most overlapping of any other inerrant species. So the Sierra yellow-legged frog uses these very deep lakes that the toads stay away from because they're too deep, they're too cold. They prefer that shallower meadow habitat, so they're kind of out of the picture. I've seen a couple of sites where they overlap barely, but it's rare. So it's pretty much the chorus frog and the toad. And I think, I alluded to this earlier, I think the reason that the toad uses those very shallow pools is not because it's good for them necessarily. I mean, shallow to an extent is good because they're gonna absorb more heat and metamorphose more quickly. But you should see some of these ponds, they're ridiculously shallow and a lot of mortality happens as a result. And so I think that's probably them competitively avoiding the chorus frog, who's kind of a generalist. They do well, they have larger numbers. And so they're trying to avoid that and find their own niche. I think they've been doing this for millions of years. And so that's why they do it. But it's pretty much just the chorus frog. Up in the north, there's the long-toed salamander. I'm not really sure how much they actually overlap in microhabitat because I haven't looked at that much. That's pretty much it.
Well, thanks. That was a great talk.
All right, thanks everyone.
Where is this toad? Where does the toad? Where does this toad end? Where does it go?
This toad, eventually she makes her way down to a meadow.
To the meadow?
Yeah, she's working her way from her hibernation spot down to the meadow so she can breathe.
It's a place where four tortoises come out, wait. And then there hadn't been any, but this one was right next to a little hole, so I just poured some of my water. And usually they just run away, but this one went.
I got it. Yeah. I warned it not to roll.
Yeah, but
I just want to say, I know, I just.
Actually offered.